Hello my dear aspirants, I hope you all are doing well. I hope you are continuing your preparations well. So again I am back with a very important topic, that is the RES map kinase pathway, which is very important for your NET and your Northeast Slet exam. So without wasting much time, let's start the video and watch the video till end and comment down below if you don't understand any specific point about that video. And also I used to give coaching for NET and Slet, so you can contact to the given number or you can comment down below if you want to study detailed syllabus from me. So let's start the video. Okay, so let's discuss about RES map kinase. So before starting RES map kinase, I just want to tell you that there are different types of receptors. In our previous lecture, we have studied about G protein coupled receptors or G protein linked receptors. But there are different types of receptor. First one is the ion channel linked or ion channel coupled receptor, which I'll discuss in some another video. Next is a G protein coupled receptor, which the basic things about GPCR I have discussed in a previous video. You can go and check out. The third type of receptors is enzyme linked receptor, and again under enzyme linked receptor there are different types of receptors, and one type of receptor is receptor tyrosine kinase. So the RES map kinase pathway, this is a pathway, okay, this RES map kinase pathway, the receptor which is involved in RES map kinase is the receptor tyrosine kinase. So here RES is nothing but this is a protein, okay, this is a monomeric G protein. And in case of GPCR also we have studied about one G protein, but that G protein was a trimeric G protein containing alpha, beta and gamma subunits. You can go and check out that previous video. But here this G protein, this RES G protein is not trimeric, rather it is monomeric. That means it has only one subunit, and that one subunit is enough to do all the required things in the signaling. The receptor, as I have already mentioned, in this entire pathway the receptor is RTK, that is receptor tyrosine kinase. The ligand here, the ligands can be of different types, but I am just giving an example that the ligand can be a growth factor. There will be some adapter protein. So here the GRB2, this protein will act as an adapter protein. Everything will be clear once we start the pathway, but here I am just giving you one introduction about the proteins that are involved in the pathway. Here we have a GEF, that is SOS. So SOS protein, this is acting like a GEF. We have a transcription activator, transcription factor activator, which is the ERK, and we have transcription factor which is FOS and JU. Let me tell you one thing, that RES, okay, this is a GTPase. That means it can switch between GTP and GDP form. So in its active form, when RES is active, that means when the pathway will be on, at that time the RES will be bound to GTP, and this is the active form of RES. When the RES is inactive, that means when the signaling or when the pathway is turned off, at that time RES will be bound to GDP. This is an inactive form of RES. When there will be ligand, then RES will be activated. When there will be no ligand, RES will be deactivated or inactive. Okay, when RES is active, then only the further signaling will continue, and when RES is inactive, then the signaling will stop or it will not be switched on. This binding of GTP with RES is done by GEF. Okay, GEF helps in binding GTP to a RES or to any G protein. And the binding of GDP with RES is mediated by GAP. GAP helps in binding GDP with RES. So here just I have told you that SOS is acting like GEF. That means SOS will help to bind GTP with RES. Let us start the signaling pathway. Suppose this is the plasma membrane of the cell, and this is the receptor. What receptor? Receptor tyrosine kinase, which is a type of enzyme linked receptor. Normally the receptors are in monomeric form, but when ligand will bind, at that time the receptor will become dimer like this. Normally they are monomeric, but when ligand will bind, at that time they will become dimer like this. Okay, because it is enzyme linked, that means some enzyme will be linked to the receptor. Here, remember the cytosolic part means this is the cytoplasm, right? Because this is the entire cell, so this will be the cytoplasm. The part of the receptor which is in the cytoplasm, that part of the receptor has enzymatic property. What type of enzymatic property? Kinase enzyme. The cytosolic part of the receptor acts like an enzyme which is a kinase enzyme. And what kinase enzyme do? Kinase enzyme, the role of kinase enzyme is to add phosphate. Okay, these kinase enzymes, it can add phosphate to some other protein, or it can add phosphate to its own body also. So this cytosolic part of the receptor is acting like kinase enzyme, that's why it is called enzyme linked receptor. So the enzyme is not a different protein. Here the enzyme is the receptor itself, but just a cytosolic part of the receptor. So when ligand will bind, at that time receptor needs to be activated, then only the further signaling will be activated. So this kinase part, or that means the cytosolic part of the receptor, will get activated. Because the receptor is nothing but a protein itself, and proteins are made up of different amino acids. Out of them, tyrosine is a type of amino acid with which the protein, this entire receptor, is made up of. So when the ligand will bind, the receptor will get activated. And how it gets activated? The tyrosine residues, because I am discussing about receptor tyrosine kinase. So the tyrosine amino acids of the cytosolic part of the receptor will get phosphorylated. So here phosphate will be added to the tyrosine amino acid of the receptor. How? First one receptor will get activated, this will help in phosphorylation of this one, again this will help in phosphorylation of this one, this will help in phosphorylation of this one. So the one monomer will help in phosphorylation of the next one, and it will help in the phosphorylation of next one. So at this stage it is known as trans So this stage is known as trans-autophosphorylation. So the first step is ligand binding, second step is trans-autophosphorylation. That means both of the monomers are helping in phosphorylating each other. Where phosphorylation is happening? In the tyrosine amino acid, because it is receptor tyrosine kinase. The receptor itself is acting like a kinase enzyme, and that kinase enzyme is phosphorylating itself only. Fine. So after the receptor is trans-autophosphorylated, so now the receptor is completely active. Now what will happen? It will help in activation of some other proteins. Now this entire part is known as phosphotyrosine domain. Okay. Now here not all protein are able to bind with this one, with this portion. Only proteins having a SH2 or PTB domain, means suppose this is a protein X, if it is having SH2, suppose this part, if this part is SH2 domain or PTB domain, then only this protein can bind with the phosphorylated tyrosine residues of the receptor. So we have a adapter protein, like I have mentioned, GRB2. GRB2 will come here, and GRB2 will bind with this phosphorylated part of the receptor, because this entire part, this part of GRB2 is SH2, has SH2 domain. Okay, because GRB2 has SH2 domain, that's why GRB2 can bind with the phosphorylated receptor. GRB2 also has a SH3 domain. With the help of SH3 domain, GRB2 will bind with another protein known as SOS. Now GRB2 will help in activation of SOS, because SOS was trying to bind with the receptor, but it was not able to bind with the receptor. Why? Because SOS has no SH2 domain. Because SOS is lacking the SH2 domain, that's why SOS needs GRB2 adapter to bind with the receptor. Now this GRB2 is helping SOS to indirectly bind with the receptor, and now SOS is very happy because now it is activated. This SOS, being a GEF, it will do what? It will help to activate RES. How? Because now it will help in binding GTP with RES. When GTP is bound to RES, see, when GTP binds to RES, then RES becomes activated. Now RES is also active. This activated RES will help in further signaling. This RES will activate one protein known as REF. So activated RES can activate REF. which is also known as MAP kinase. Activated REF can help in activation of another kinase which is known as MEK. This MEK is also known as MAP kinase. Activated MEK can activate another kinase which is known as ERK, which is also known as MAP kinase. So REF, MEK and ERK, all three are kinase enzymes, and REF activates MAP by adding phosphate in the serine or threonine residues. MEK activate ERK by adding phosphate in the serine or threonine or tyrosine residue, and ERK can help in activation of transcription factors. Okay, ERK help in activation of transcription factor in the nucleus, okay, which is called June. ERK help in activation of transcription factor of phos and June, and when they are active, when phos and June is active, then it will go and it will sit upon specific gene of DNA. Phos June will help in activation of such gene of DNA. It will help in transcription of such a gene which will make a protein in the cytoplasm, and this particular protein will be some growth factor. Okay, so I am repeating it once again. First, ligand will bind. After ligand binding, receptor will get activated. How? Its enzymatic part will get activated, and then trans-autophosphorylation will happen. After trans-autophosphorylation, all the tyrosine residues of the cytosolic part of the receptor will get phosphorylated, and then one adapter protein having SH2 domain, which is known as GRB2, will bind with the phosphorylated receptor. GRB2 has another domain which is called SH3 part or domain. With the help of SH3 domain, it will bind with SOS. SOS is acting like a GEF, so it will add GTP to the RES. Upon addition of GTP, RES will get activated, and RES will activate REF, which is MAP kinase. REF, because it is a kinase enzyme, so it will add phosphate to one another kinase known as MEK. Okay, after addition of phosphate, MEK will get activated, and again MEP will activate another kinase known as ERK. ERK is also a kinase enzyme. It will activate phos and June. Phos June are transcription factor. Phos and June will go and sit upon DNA, and then it will help in transcription, means from this particular gene one RNA will be formed. This RNA will make one protein. That protein will be a growth factor. So this ligand was coming with an information that now growth factor has to be made. So this ligand is finally helping in activation of a growth factor. This is how RES-MAP kinase works. So this entire pathway is known as RES-MAP kinase pathway. I hope you understood the video. If the video is helpful enough, then don't forget to share it with your friends. And if you want to take detailed coaching of Slet, then you can contact in this given number. Okay, I provide full detailed course of Slet coaching. Bye bye. Please subscribe, and all the best for your upcoming exams.